Okay, I guess we're all here. Let's get started. Everybody have a culture inoculated now for your known for Wednesday? Yeah, raw culture. Not going to do that. Tomorrow we're going to do several things. We'll do the motility tests. There are 2 motility tests we'll do. We will start a medical microbiology exercise, which will carry over for a couple of weeks. And then I also want to go through some things about the lab exam. The lab exam is coming up, and I'd like to give you some heads up about what kind of things you are responsible for and what kind of things you should be preparing for. So we'll do that on Wednesday too. Any questions?
What day is our pure culture, mixed culture?
That's due by the lab exam. Okay, that's due the day of the lab exam, or before if you want to give it to me. Okay? Okay, let's go back and do a little kind of step back for a second as we're talking about these various control methods. We're going to talk about and are talking about 3 major categories: the physical control methods, mechanical and chemical control methods. We spent quite a bit of time talking about the effect that temperature can have on the growth of microorganisms, and we spent all of our time really talking about heat and differentiating between moist heat and dry heat. And so dry heat would include things like putting things in ovens for long periods of time, incineration, as you do with your inoculating loops, and then the various moist heat methods that will include boiling, which will disinfect but not sterilize, pasteurization, which will also disinfect but not sterilize, and then autoclaving, which will give us our true sterility. I didn't say anything about cold before I moved on to radiation, and I should have. And so let's talk for a second about cold. What effect does cold have on controlling the growth of microorganisms? It slows down its metabolism, okay? That's why we put things in the refrigerator after we have incubated them. You incubate to get maximum growth. You're putting those organisms in their ideal temperature. So we want cell division to be occurring as fast as it possibly can. But then once it's growing, we want to maintain those cultures by putting them in the refrigerator and slowing them down. What about if we stick them in the freezer? What's that going to do? It's going to slow down their growth even more, right? But will it have any lethal effect? Potentially something could start producing endospores. When you freeze something, water undergoes an interesting transition. And as it turns from a liquid to a solid, what happens to it? It expands and crystals form. And so one of the reasons why freezing things actually is destructive to cells is that the water that's present in the tissue expands as it freezes and forms ice crystals. And those ice crystals, if they form within a cell, literally tear the cell apart. And so freezing can have some lethality to microorganisms because of that particular feature. Now, if we freeze rapidly and under a vacuum, water does something interesting. As it changes state, it doesn't form those ice crystals. And cultures that are rapidly frozen under liquid nitrogen, for example, under a vacuum, change pretty much immediately from that turbid broth liquid solution to a freeze-dried powder. And as that transition takes place from liquid to a solid, this doesn't happen. This particular process is called lyophilization. And this actually is used as a way to preserve bacterial cultures for long periods of time. In that freeze-dried lyophilized state, those cells are still viable, but they're pretty much completely inert. They've been completely dehydrated. You can maintain in a regular freezer or even refrigerator freeze-dried cultures of bacteria literally for decades. And what do you do to reanimate them? Add some liquid. So you can take a little bit of that powder out and sprinkle it into a tube of broth, and those cells rehydrate, and all of a sudden, bingo, you've got a new viable culture. Moving over to radiation, we looked at the electromagnetic spectrum and distinguished between the more intense forms of radiation on the left of the spectrum, gamma rays, X-rays, shorter wavelength, more penetrating, more damage to cells, and contrasted that with the longer wavelength forms of radiation, including ultraviolet radiation. A little bit longer wavelength, less damaging. The shorter wavelength, we said, were considered to be ionizing forms of radiation, and the longer wavelengths of radiation were both considered to be non-ionizing. Both have the potential to be lethal, this one more so than this one. But they have different mechanisms. This one works primarily by breaking molecules apart, forming a whole lot of free radicals, which are highly damaging to cellular structures, where the mechanism of non-ionizing radiation is quite different. That involves the formation of these abnormal bonds in DNA, the thymine dimers. We'll come back and look at that in a little bit in just a minute. But let's take a look at the ionizing radiation for a second. We already talked about this a little bit in the context of food treatment. A lot of food is lost due to spoilage, and also food can be a potential source of pathogenic organisms which can cause serious disease. So I mentioned before there are several different types of bacterial food poisoning. And remember, that term food poisoning is kind of a generic term. The exact terms when you're talking about foodborne illness are actually food intoxication. Over on the left. And then intestinal infection or food infection caused by living cells. And so the intoxication is produced by toxins produced by cells, which may be dead now. Food infections are caused by living, multiplying microorganisms. But there's a pretty long, impressive list of these diseases, and they range from very annoying to lethal. So one of the things that we want to do, and this is the province of the Food and Drug Administration, is make sure that the food supply chain in the food industry takes proper steps to try to ensure that foodborne contamination does not cause disease outbreak. One of the methods that's traditionally been used to prevent food contamination would be proper heating of food. Salting of food was also traditionally used, in meat preservation particularly. But one of the areas of real promise is using forms of radiation, including ionizing radiation. And those fall under the category basically of food irradiation. The most common form of ionizing radiation that's used to irradiate food is gamma radiation. There are machines called cobalt irradiators which produce gamma rays in a very controlled fashion that can be used in the food industry to irradiate food. How effective is it? Well, here you look at strawberries. On the left, those strawberries have not been irradiated, and these are irradiated strawberries that have been around under the same conditions for the same amount of time. And you can see that obviously here, the decomposers are having a great time breaking down the tissues of that particular fruit. Over here, the irradiation has killed those decomposers, and the fruit is much more stable and has a much longer shelf life. There are, a pretty impressive list of foods that can be legally irradiated, and this particular list is growing longer and longer and longer. One of the things that you will sometimes see in stores that sell irradiated food are these green labels right here that indicate that the food has been irradiated. This is a consumer protection and knowledge requirement, because the Food and Drug Administration feels that consumers have the right to know that this particular food has been exposed to irradiation. Arthur, jump in.
Is it completely safe?
Okay, that is the question. Is it completely safe? Some people say absolutely, and that is the scientific consensus, but there is real lingering fear out there that food irradiation is somehow harmful to the people who ingest the food. There is no lingering radiation associated with the food following food irradiation. Okay, so the food is not radioactive. Okay. The only concern at all is that irradiation does produce free radicals. And those free radicals are part of the killing mechanism that destroy bacterial and fungal contaminants within the food. Those free radicals have very, very short lifetime. And all of the scientific evidence to date indicates that those free radicals dissipate very quickly, and that irradiated food is completely safe and harmless. Nevertheless, truth in advertising, the radiation symbol has to be associated with foods that are preserved in this particular fashion. The savings, both monetarily and health-wise, associated with widespread irradiation of food are pretty impressive. And there's a couple of figures in the lecture notes that are just there for illustrative purposes. I don't expect you to memorize those, that indicate the savings that could occur both in the food industry in terms of loss of food and in terms of healthcare costs for preventing disease. Why aren't we doing more of this? Because of public concern about the effectiveness and safetiness. Consumers have to buy into the fact that this is not going to harm them, or there's not going to be widespread public acceptance of this. There's a lot of applications for irradiation, which are very, very intriguing, particularly in less developed countries, because the primary method of food preservation worldwide right now is refrigeration. And refrigeration is costly. It requires electricity. On the other hand, these gamma irradiators are portable. You don't need a widespread infrastructure where every house has a refrigerator, for example. And so small processing units could be set up, even in remote areas, where the food is brought to these particular facilities and radiated and then distributed out to households where the need for refrigeration would be either eliminated or dramatically reduced. So there's a lot of promise in this, but there's a lack of public acceptance because of the word radiation.
How long would they, once they're irradiated, would they just then be available to be contaminated in?
Right, I mean, these don't have an unlimited shelf life. This is something that will just extend the shelf life. But yeah, once they pick up contaminations following radiation. Now, if you package them properly when you do the irradiation, and they can't get exposed to air contaminants or soil contaminants, you're going to have a pretty long shelf life. But it's not a matter of them now being sterile. It's just a matter of extending the lifetime of these particular food resources.
What if you have radioactive resistant bacteria?
That would be a concern, okay? And remember that we said that endospore-producing bacteria have about a double increase in the resistance to radiation than non-endospore-forming bacteria. But if you adjust the intensity of the radiation with the time of exposure, you can even kill those resistant endospores. Now, one of the things you've got to be concerned about is, does this irradiation affect flavor, texture, and other kinds of things that might make it unpalatable to a consumer? Because if this stuff tastes different to a consumer, consumers are not going to buy it. Okay? So it's really kind of like the heat treatment. You've got to find the right conditions which destroy the contaminants but don't really alter the palatability of the food that's being irradiated. So, you know, there's some work to still be done, but there's some real promise in the food industry of increasing irradiation, if the public will accept it. Okay, ultraviolet radiation is a different beast. Ultraviolet radiation is longer wavelength, lower energy. And when molecules absorb that ultraviolet radiation, they don't split apart. You don't form all of these destructive free radicals. But the energy can be absorbed by critical molecules like DNA and form abnormal bonds that weren't there before between adjacent thymines on the same DNA strand. And that's called a thymine-thymine dimer. And I told you last time that these dimers interfere with both DNA replication and with transcription. So that's not good. Now, bacteria do have repair mechanisms to create mistakes in DNA. And one of the things that those particular repair mechanisms typically do is to cut out damaged segments of DNA. When they cut out the damaged segments of DNA, they don't just cut out this particular dimer, they cut out this particular whole section right here. Okay.
the lecture.
Good point. I would have stopped it, but then I probably would have started it again.
Oh, no, yeah, right.
Uh-oh.
He probably wants to bring it up.
Okay, that gave you a little bit of a wake up there. So anyway, as these particular damaged segments are cut out, you're removing your template strand. And so when it comes time to repair that, what are they going to stick in there? There's no template to dictate what the complementary strand is going to be. So very oftentimes a mistake is introduced as a result of that kind of crude repair mechanism. So one of the mechanisms by which ultraviolet radiation kills is that it induces a high frequency of mutations following this repair process, and those mutations can oftentimes be lethal. Ultraviolet radiation has a limitation, and one of the big limitations that it has is that it has poor penetrating power. So it can be good for disinfection of air, for example, but it has much less utility in terms of disinfecting a liquid or something dense like a food, for example. Where do we see these ultraviolet lamps that are typically used to try to disinfect air? Well, one of the places we see them is in structures called transfer hoods. I don't know if you've ever wondered what this thing is up here. This is an old-time transfer hood. This is used to try to create a less contaminated environment for transferring cultures. You can just put it on the tabletop, for example. And then if you wipe the tabletop down with disinfectant, then at least the floor of the transfer hood is fairly germ-free. And then what you do is there's an ultraviolet light up in the top of this transfer hood, and then you flip that on for about a half an hour ahead of time, and then that will tend to disinfect the air and create a more germ-free environment for transferring your cultures. I'm going to show you a laminar flow hood in a second. That's based on another kind of technology involving a mechanical control method. But laminar flow hoods also have ultraviolet lights built into them to try to enhance the chance of getting a fairly germ-free environment within that container. You'll find these ultraviolet lights sometimes in public restrooms. I noticed that in Europe when we were there a few years ago, and that they had these UV lights high up in the public restrooms to try to sanitize the air a little bit. They can sometimes be used in hospital settings, in operating rooms, and pre-op wards. Just the intent is to try to reduce the microbial load to prevent less chance of contamination. We're going to talk about water treatment later on when we talk about chemical control of microorganisms. What's the standard chemical compound used to treat water to try to kill bacteria? Come on, swimmers. Chlorine. Chlorination, okay. And chlorine is a very effective disinfectant for water. Works really well. We'll talk about why it works. But there are some downsides to chlorine that we'll talk about. One of the things that people have oftentimes kind of thought about is, could we use ultraviolet treatment of water as an alternative to chlorination, because ultraviolet treatment doesn't have some of the bad side effects that chlorination can have? And the answer is yes, but you've got to set up your water treatment system in a very specific way. And one of the things that you have to do is when water passes through an ultraviolet treatment in the water treatment process, it's got to be in a very thin film, so penetration of the UV into the water is not an issue. And so this is an advanced water treatment plant with ultraviolet treatment set up, and the water is passed through these ultraviolet lights or over the ultraviolet lights in a very thin film, allowing the poor penetrating ultraviolet radiation to kill microorganisms that are present in that water. This typically is a final step of water treatment. There are other forms of water treatment that occur before this final step of either chlorination or ultraviolet treatment. I don't know if anybody's ever seen this before. This is a little ultraviolet sterilizer for water called a SteriPen. And this is something you can use if you go to a foreign country where they say you really shouldn't drink the water because it might be contaminated with microorganisms, especially ones that are not part of your normal flora. Sometimes you get sick when you go to another country because you are being exposed to organisms you're not normally exposed to. They're just not part of your body's normal microbiota. These SteriPens are kind of cool. What you can do is you can put them in a glass of water or a bottle of water. You turn it on, they're battery operated, or some of them have little solar chargers with them, and you just swirl it around in there for about 30 seconds, and the ultraviolet light then kills the bacteria and makes the water fit to drink. So these are pretty cool devices if you travel a lot. Some of you, I don't know if any of you use electric toothbrushes. We just got one, and our electric toothbrush has a little device for ultraviolet treatment of your toothbrush to try to reduce the bacterial load on your toothbrush. Okay, let's switch over to mechanical control, another physical control method. The major type of mechanical control we'll talk about, there will actually be two of them. The major one we're going to talk about is filtration. And I know some of you have worked with Dr. Chiquan on the water project, and I know that there has been some filtration that has been used in bacteriological water testing. One way in which you can create microbe-free solutions is to pass them through a filtering apparatus, and a membrane filter separates the non-sterile material from sterile material that's collected by filtration. Usually this is coupled with hooking this collecting vessel up with a vacuum pump to pull the liquid through the filter more rapidly. If you had to rely on gravity, it would be very slow for that liquid to drip through that filter. The basis of this particular filtration is the fact that the filter material, which is typically a cellulose compound, cellulose acetate, has little holes in it of a defined diameter. And you can get cellulose acetate filters of all different kinds of pore size. If you are trying to make sure that you have no bacterial contaminants in your collected material, then the pore size can be a little bit larger than if you want virus-free solutions, for example. Then the pore size is going to have to be smaller. The problem with small pore size is that even under vacuum, it can be tough to pull liquids through very small holes. But the idea is that any contaminants, in this case bacterial cells, are trapped on that filter media because they're too large to pass through the small pores in the filtering material. There are some bacteriological samples that just don't autoclave well. Media that contain sugars oftentimes are heat sensitive and can turn into a real nasty mess if you heat them in an autoclave. That process is called caramelization. And I don't know if you've ever just cooked sugar in a recipe, but if you overcook sugar, you can just get this mass of kind of sticky material. That's what happens to media sometimes that contain sugars. So this membrane filtration can be an effective way to sterilize the media without heat. One of the media that we're going to use later on in class for a test called the urease test is to see if your microorganism has the enzyme urease, which can break down the compound urea into ammonia and CO2. So we need to make bacteriological media with urea in it. Well, if you autoclave urea, it automatically decomposes into ammonium and CO2. So that's self-defeating. So when I prepare this particular media, I have to membrane filter that urea solution to make a sterile urea solution, and then I can aseptically add that to agar and make these urea agar plates that we'll use in the lab. So this has very strong applications in filtering liquids. There is a organic material called diatomaceous earth, which also is used in membrane filtration. It's typically used for larger scale filtration. You can get diatomaceous earth filters for filtering aquarium water, and you can also get large diatomaceous earth filters for swimming pools. But again, the idea is that the diatomaceous earth will trap the contaminants, allowing the water to pass through, and the contaminants would be held behind. It's just a different mechanism. Diatomaceous earth uses more of a chelation type of phenomena, where these membrane filters use just pore size as the filtering mechanism. Now, we not only can filter liquids, but we can filter air. And this is a biosafety cabinet. The purpose of this biosafety cabinet is to pull air through the cabinet and do so in such a way that the user is not exposed to any noxious substances that might be present in the samples that you're working with. We don't have biosafety cabinets, but we do have something called a laminar flow hood. And a laminar flow hood is designed to try to filter air so that we create a fairly sterile environment that protects the samples that we're working with. Have any of you worked with Dr. Rhodes on his tissue culture project where he does the cow oviduct cells and environmental estrogens and things like that? Nope. Okay. Well, in tissue culture, you need to create an environment that doesn't allow contamination of the cells that you're trying to culture. And that laminar flow hood basically is set up to filter the air using these particulate filters called HEPA filters, which draw in contaminated air, trap the contaminants like bacteria and fungi, and then only allow filtered air to pass through the cabinet where these open dishes are that you're working with. Okay? So HEPA filtration is something that is used to filter air. Laminar flow hoods also typically have ultraviolet lights associated with them. That just augments the ability to create a fairly sterile environment inside of these enclosed hoods. Sometimes the HEPA filters are also impregnated with antibacterial chemicals, which improves their efficiency. You're now seeing HEPA filtration marketed for home furnace air filters, particularly for people who have real allergies. You also will see sometimes HEPA filtration advertised on vacuum cleaners right now, so that the exhaust air coming out of the vacuum cleaner doesn't just re-add pollutants back to the air again from the stuff that you're vacuuming up. So HEPA filtration primarily for air filtration. Well, I mentioned the food industry when we talked about irradiation. Within the food industry, there are several different points at which bacterial contamination could be introduced. And so typically, working in the food industry involves employing several different methods of control at different points throughout the cycle of preparing and processing that particular food. And it includes things like use of heat, cooking, pasteurization, canning, the use of radiation. If it is a liquid food or beverage, filtration is widely used. We don't pasteurize liquids nearly as much as we used to anymore. Beer used to be pasteurized a lot. Now you'll hear some beer companies advertising that their beer is cold filtered, for example, and that is a filtration mechanism to remove contaminants. Wine is more commonly now filtered to remove yeast sediment and things like that than pasteurization. Proper maintenance of temperature, either for storage or for cooking, is crucial in the food industry. And then another thing that's very important and prevalent in the food industry is the use of chemical additives. So that's kind of where we're going to go next, is talk about some of the chemicals that can be used to preserve food by killing and arresting the growth of microorganisms. So this is just a good summary slide again of some of the physical methods of microbial control. Again, numbers are not important right here. I think probably the one number you should remember, maybe two, is that boiling water, 100 degrees Celsius, doesn't kill endospores. It will disinfect, but it won't sterilize. And I think the other number probably that's important to remember is, what temperature do you achieve in an autoclave at 15 pounds per square inch? That 121 degrees Celsius. And understanding that extra 21 degrees Celsius is the difference between killing an endospore and not killing an endospore. But in terms of how many seconds does it take to ultra pasteurize versus flash pasteurize, all of those details are just kind of illustrative examples. One of the things that you will see is that a common denominator of action is that many of these particular treatments work by causing protein denaturation. Here, some of the cold temperatures inhibit metabolism. Some methods involve interacting with and making DNA molecules damaged or non-functional. But protein denaturation is really one of the major mechanisms. And you're going to see that's going to be the case also when we talk about chemical destruction of microorganisms. One of the major ways that chemicals destroy microorganisms is by denaturing their proteins. We typically call chemicals that can kill or inhibit the growth of microorganisms germicides. And that's kind of a generic term. A germ is kind of a catch-all phrase to denote something that can cause disease. Germs can be bacteria, they can be fungi, they can be virus. Anytime you start getting chemical compounds involved, and particularly if human tissues are also involved, then the whole issue of toxicity becomes really important. And that's particularly important if you take a chemical internally. Because it's one thing to expose the surface tissues of your body to a chemical compound that might be a little bit toxic to cells. Yeah, it might kill some skin cells, but that could be a good trade-off. Sacrificing some skin cells, which are renewable, for killing pathogens which could cause serious disease. But you start taking things internally, and it's a whole new ballgame. Because your internal organs can then all be exposed to these chemicals, particularly if they're transported around by the circulatory system. So once we talk about internal compounds, this idea of selectivity is really, really important. Can we find something that will target the pathogen, but leave human tissues alone, like an antibiotic, for example? But we're not going to talk about that on this exam. Instead, we're going to talk about chemical compounds, which are primarily intended for external use only. What do we call chemical compounds that are acceptable to use on the external surface of tissues? Antiseptics. Okay? Ones that are usable on inanimate objects but not external tissues are the disinfectants. But the disinfectants are a little bit harsher, more damaging. So if you were to sit down and kind of build the perfect germicide, what kind of characteristics should it have? Well, it would be nice if that particular germicide didn't have to be present in really high concentrations. Because typically speaking, any chemical compound, its toxicity increases with its concentration. So the lower the concentration you can use and still be effective, kind of the better off. These compounds are ideal if they can be water or alcohol soluble. It's hard to make a solution of things for dissemination, for a spray, for example, if it can't dissolve. Things that are alcohol soluble are called tinctures. You may have heard of tincture of iodine before. That is a solution of iodine dissolved in alcohol. Chemicals that have a broad spectrum, meaning they can kill a wide variety of different types of organisms, are more desirable, particularly if they don't have human toxicity. If they have better penetrating power, that's good. Remember I said that organic matter, like dirt, can be a chelating agent. And so if compounds are quickly chelated by organic matter, that can be problematic, particularly if you're intending the substance to be a cleanser or a cleaner. Expose it to dirt and it's inactivated, that's not good. Non-corrosive and non-staining, that's one of the drawbacks of iodine. Iodine tends to turn things brown. And sometimes that's okay, other times not. Cost is always an issue. People probably aren't going to use something if it smells horrible. So that's a pretty long list of things. Very few chemical compounds achieve most of those goals. Probably the two that come closest to being an ideal germicide and hit most of those things are hydrogen peroxide in very high concentrations. Not the stuff you can buy in the bottle at the drugstore. That's not high enough concentration. And there's another compound we'll talk about called chlorhexidine. Those are two compounds that kind of hit most of these benchmarks and are considered to be fairly ideal germicidal agents. One of the first categories of compounds that was used in chemical disinfection goes back to Lister, who you might remember from the beginning of the semester, was a surgeon who discovered that if you disinfected surgical instruments, your rate of surgical infection dropped tremendously. Lister washed surgical instruments in his hospital in a solution of a substance called carbolic acid. Carbolic acid is another name for the compound phenol. So here's phenol right here. Little aromatic ring with a hydroxyl group attached. Pretty innocuous looking compound. But quite antibacterial. Phenol is a very potent denaturer of proteins. It also has an inhibitory effect on cell membranes. And it can cause just kind of dissolution of cell membranes, breaking down of cell membranes. Phenol is kind of a nasty chemical. It can cause serious chemical burns. If you get some phenol on your skin, it can cause blistering and real destruction of the tissues, which gives you some kind of an idea of how toxic it is. Because it was one of the first chemicals that was used, phenol has become kind of a benchmark for comparing the effectiveness of one chemical to another chemical in terms of its killing ability. You can calculate something for a chemical called the phenol coefficient. And basically what this coefficient does is it says, this chemical kills this much relative to phenol. So here is the phenol coefficient of some common chemical compounds that we use in antisepsis and disinfection. And you'll see that some compounds are moderately effective compared to phenol. Look at formalin, preservative that's used in embalming fluid, used to be used in dissected specimens. It only has 0.3 compared to phenol's coefficient of 1. But this compound chloramine, 133 times more effective than phenol. Mercury chloride, 100 times more effective. And so this particular comparative scale is oftentimes used to compare the relative effectiveness of different chemical compounds. Phenols are sometimes found in natural oils and are the reason some of these natural oils are antibacterial. Pine oil, for example, contains natural phenolic compounds, and some cleansers like Pine Sol is a stuff you can use to wash your floor, contains these natural pine oils with these phenolic compounds. Phenols have been chemically modified by the addition primarily of methyl groups to be more effective in disinfection and more effective as cleaners. These chemical modifications, which form some of these derivatives of phenol, are more stable in the presence of organic matter and are a little bit less toxic, particularly to human tissues. So some of these derivatives of phenols that involve addition of methyl groups, those derivatives are called cresols. One naturally occurring cresol occurs in a substance called creosote. And creosote is a black, tarry compound. If you've ever seen a telephone pole that's been dug up, it's oftentimes black at the bottom. That's because it's been coated with this creosote material. And the intention of that is creosote contains some of these natural cresols, and therefore they hope that the host will decompose less in the soil if it's coated with this antibacterial compound. Another chemical modification of phenols is to stick two of them together. That forms a group of compounds called bisphenols, bi-2. This is a common bisphenol used in chemical cleaners, orthophenylphenol. This is one of the active ingredients in Lysol. Lysol also contains some of these cresols, so it's kind of a mixture of different types of phenolic compounds. This bisphenol is an interesting compound, hexachlorophenol, and that's probably where we need to pick up next time. We'll finish up cresols. We do have some more work to do on the rest of the chemical compounds, but we will get through those tomorrow, or Thursday, sorry. And that will be the ending of the exam material. So make sure you have your culture of your unknown.